Hello and welcome to our new topic on antibacterial drugs that work by inhibiting bacterial cell wall synthesis. These drugs are among the most important and most frequently prescribed antimicrobials in medicine today. They include penicillins, cephalosporins, carbapenems, monobactams, vancomycin, and several other agents commonly used in clinical practice across all specialties. Throughout this lecture we will build that knowledge step by step, beginning with the bacterial cell wall itself and ending with the major drug classes that target it, and a summary at the end. So, let's start. The learning objectives of this lecture series are listed here. Please take a look. Antibacterials remain among the most frequently prescribed drugs worldwide. This graph illustrates that cell wall synthesis inhibitors alone represent an overwhelming majority of the global market. Such widespread use has transformed medicine by dramatically reducing mortality from infectious diseases. The SOFI profile, antibacterial spectrum, availability of oral dosage forms, cost-effectiveness, and species selectivity are among the important factors why cell wall synthesis inhibitors are so widely and commonly used today. That doesn't mean other groups are unimportant, and this we will discover as we complete our journey through the fascinating world of antimicrobial drugs in this lecture series. To understand cell wall inhibitors, it is helpful to first review the overall structure of a bacterial cell. The bacterial chromosome contains genetic material, while plasmids often carry resistance genes. The cytoplasm is enclosed by a cell membrane, and surrounding that membrane is the bacterial cell wall. The cell wall provides structural integrity and protects the organism against osmotic lysis, because it is the outermost covering. Unlike mammalian cells, bacteria possess this unique structure, making it an ideal target for selective toxicity. Antibacterial drugs can therefore damage bacteria without harming human cells because human cells lack peptidoglycan cell walls. Several bacterial structures and biochemical pathways can be targeted by antibacterial drugs. Some drugs interfere with nucleic acid synthesis, others inhibit protein synthesis, and some disrupt cell membrane function. The focus of this lecture is the peptidoglycan cell wall. Because the cell wall is essential for bacterial survival and absent in human cells, it represents one of the most successful targets, showing species selectivity in antimicrobial therapy. Many of the most widely used antibiotics exert their effects through interference with cell wall synthesis. The bacterial cell wall differs significantly between gram-positive and gram-negative organisms. Gram-positive bacteria possess a thick peptidoglycan layer and contain teichoic acids. They do not have an outer membrane. In contrast, gram-negative bacteria possess a much thinner peptidoglycan layer but are surrounded by an outer membrane that contains lipopolysaccharide, or LPS. This outer membrane serves as an additional permeability barrier and significantly influences antibiotic susceptibility. These structural differences explain why some antibiotics are highly effective against gram-positive organisms but less effective against gram-negative bacteria. Cell wall synthesis is a dynamic process that occurs continuously as bacteria grow and divide. In cocci such as Staphylococcus aureus, synthesis is concentrated at sites of cell division. In rod-shaped organisms such as Bacillus subtilis and Escherichia coli, synthesis occurs both during elongation and division. Because bacterial survival depends upon constant cell wall remodeling, interference with these processes can rapidly lead to cell death. Cell wall synthesis can be divided into four major processes. The first is cytoplasmic synthesis of peptidoglycan precursors. The second involves membrane transport of these precursors using a lipid carrier known as bactoprenol. The third process is transglycosylation, during which glycan chains are assembled. The final process is transpeptidation, which cross-links peptide side chains and provides strength to the cell wall. Different antibiotics act at different stages of this pathway, and understanding these stages is crucial for understanding their mechanisms of action. Several important antibacterial drugs target distinct stages of cell wall synthesis as shown. Phosphomycin acts very early during precursor synthesis. Cycloserine interferes with D-alanine metabolism. Bacitracin disrupts recycling of the bactoprenol carrier molecule. Vancomycin binds peptidoglycan precursors and prevents their incorporation into the cell wall. Finally, beta-lactam antibiotics inhibit transpeptidation by binding penicillin-binding proteins. Together, these drugs demonstrate how multiple points within a single pathway can be exploited therapeutically. The major drugs that inhibit bacterial cell wall synthesis can be divided into two broad categories. The first and most important category consists of the beta-lactam antibiotics, including penicillins, cephalosporins, carbapenems, and monobactams. These agents share a common beta-lactam ring and primarily inhibit transpeptidation by binding penicillin-binding proteins. The second category includes structurally diverse agents such as vancomycin, teicoplanin, bacitracin, cycloserine, and phosphomycin. Although these drugs differ in chemical structure and specific targets, they all ultimately interfere with cell wall formation and weaken the bacterial cell wall. Understanding this classification provides a useful roadmap for the remainder of the lecture. Many antibiotics, including those discussed in this lecture, originated from microorganisms themselves. This slide shows the beta-lactam antibiotics, whereas others are listed in the next slides. It is important to note that the original antibacterial molecules later used to develop many semi-synthetic and synthetic drugs in that class. For example, penicillin G is the original microbial-derived molecule that was used to generate all other penicillins. The non-beta-lactam drugs are listed F here with their sources. It is noteworthy that while beta-lactam drugs evolved into a long list of compounds which have become most commonly used antibacterials, the non-beta-lactams largely remained as such original molecules. The beta-lactam antibiotics constitute the largest and most important family of cell wall synthesis inhibitors. Their defining structural feature is the beta-lactam ring, a four-membered cyclic amide that is essential for antibacterial activity. Despite considerable differences in spectrum and pharmacokinetic properties, penicillins, cephalosporins, carbapenems, and monobactams all share this common structural element. Notice that clavulanic acid also contains a beta-lactam ring. However, unlike the other compounds shown, clavulanic acid has very little intrinsic antibacterial activity. Instead, it functions as a competitive beta-lactamase inhibitor when used with a beta-lactam drug and thus protects susceptible antibiotics, such as amoxicillin, from enzymatic degradation. The beta-lactam ring is therefore both the source of antibacterial activity and the target of many bacterial resistance mechanisms. To understand the action of beta-lactams, it is important to appreciate how bacterial cell walls acquire their strength. Peptidoglycan consists of long glycan chains connected by peptide cross-links. These cross-links act like reinforcing beams within a building, providing rigidity and structural stability. The enzymes responsible for creating these cross-links are known as transpeptidases, or penicillin-binding proteins. Beta-lactam antibiotics bind to these enzymes and inhibit their activity. As a result, peptide cross-linking cannot occur. The cell wall becomes progressively weaker, particularly during growth and division, eventually leading to osmotic instability and bacterial lysis. Because of this mechanism, beta-lactams are generally bactericidal agents. Penicillins are the oldest beta-lactam antibiotics and remain among the most widely used antibacterial agents. They can be classified into several groups according to their spectrum and resistance to beta-lactamases. Natural penicillins such as penicillin G and penicillin V are highly effective against streptococci, enterococci, and certain other susceptible organisms. Long-acting preparations such as procaine penicillin G and benzathine penicillin G provide prolonged therapeutic concentrations and are particularly useful when sustained exposure is desired. Anti-staphylococcal penicillins such as cloxacillin, oxacillin, and nafcillin were developed to resist staphylococcal beta-lactamases. Aminopenicillins such as amoxicillin and ampicillin possess broader gram-negative activity, while anti-pseudomonal penicillins such as piperacillin extend activity against difficult gram-negative pathogens including Pseudomonas aeruginosa. Understanding these subclasses allows us to predict spectrum and clinical applications. Although penicillins are generally safe, several adverse effects deserve attention. The most important are hypersensitivity reactions, which may range from mild skin rashes to life-threatening anaphylaxis. These reactions occur because penicillin degradation products can act as haptens and trigger immune responses. Superinfection may occur when normal flora are disrupted during therapy, allowing resistant organisms to proliferate. Intramuscular administration may produce pain and sterile inflammation at the injection site. Less common adverse effects include granulocytopenia and hepatic dysfunction, particularly with certain anti-staphylococcal penicillins. Some penicillins can impair platelet function and increase bleeding tendency. Neurotoxic manifestations such as headache, dizziness, and seizures may occur with certain formulations, particularly at high concentrations. This slide summarized the entire lecture in terms of their mechanism and clinical implications. This summary slide integrates many of the practical concepts discussed throughout the lecture. It compares major penicillins according to clinically relevant characteristics such as MRSA coverage, anti-pseudomonal activity, oral availability, cerebrospinal fluid penetration, bone and joint penetration, and common therapeutic uses. We have now completed our discussion of antibacterial drugs that inhibit bacterial cell wall synthesis. Throughout this lecture, we have examined the structure and importance of the bacterial cell wall, reviewed the major stages of peptidoglycan synthesis, and explored the diverse drugs that target different points within this pathway. Thanks for watching. Don't forget to like, subscribe, and share. Happy learning!